Welcome to e-learning science. I'm Dr. Mudasir, and in this video tutorial we'll be discussing sex determination methods in case of humans, birds and honeybees. First of all, what is sex determination? Immediately a question comes in our mind: what is sex determination? Sex determination means establishment of male and female sex. If we establish this person is a male, if we establish this person is a female, establishing a male and female involves sex determination. Now the question is, what are the factors which are responsible for establishing a male, and what are the factors which are responsible for establishing a female? There may be some important factors that may be establishing a male and that may be establishing a female. See, if we see in case of different organisms, in case of humans, birds, honeybees, we have number of methods which is particular to a particular species by which we can determine the sex of any particular species. When we see sex determination, we have several methods. Several methods are available in this sex determination. The first method of sex determination is the or we can say environmental method of sex determination. The second method is non-allosomic method of sex determination. The third is hormonal method. Hormones may be determining the sex. Hormonal method of sex determination. And the fourth is haploid-diploid method of sex determination. Here the haploidy and diploidy may tell us what is going to be the sex. And the last is the chromosomal method of sex determination. We have so many methods, several methods. When we talk of different organisms, we have different methods by which can establish the sex of a male and a female. We will discuss them. Some methods may be in humans, some of the method may be in the birds, some may be in honeybees. We will discuss them one by one. Just the most important and the major method which determines the sex of number of individuals is the chromosomal method of sex determination. Before going to this chromosomal method, let's discuss a little bit of these other methods also. The first method is environmental method. Environmental method means here the environment is going to determine the sex of any individual. I will tell you this way. For example, we have turtles. If you see in case of turtles, if the temperature is less than 28 degrees centigrades, the eggs will develop into males. And if the temperature outside is greater than 33 degrees centigrades, then there is more probability we may get more and more females. And if the temperature is in between, it means it is between 28 to 33, we'll be getting both males and females. The sex ratio, whether it is the establishment of male, whether it is the establishment of female, is determined by the environment, and particularly here in this case, it will be determined by the temperature. Similarly, we have a marine worm known as Bonilla. This Bonilla is a free-floating larva, and the larva has no sex. It is free-floating. If the larva gets fixed, it becomes female. And if another larva, which will be moving around, comes near to the female, it develops into male. This is what is happening in Bonilla. So this is the first method, environmental method. The second method of sex determination, is non-allosomic non-allosomic method of sex determination. Non-allosomic method of sex determination. See, normally, when we see, usually, it is the allosome which determines the sex. But if in any case, the allosome is not determining the sex, but it is being determined by some other chromosome, maybe an autosome, we can suggest this is non-allosomic. That method we can suggest as non-allosomic. For example, it means here some of the autosome will be determining the sex, but not the allosome. Sex will be determined by the autosome, not by the allosome. In bacteria, we have F positive. We have two types of bacteria: F positive and F negative. Okay, and we can say the F positive and F negative thing is governed by the gene present in the plasmid. If somebody has F plasmid, we can say it is F positive. If somebody do not have, it is F negative. And we say this plasmid gene is not allosomic gene. It is non-allosomic gene, which is determining the sex in case of bacteria, and we call it as non-allosomic method. Similarly, we have seen in case of males, the sex chromosomes are absent, but we have the autosomal genes, and one of the autosomal gene will determine the formation of male kernel, and one of the male inflorescence, and one of the autosomal gene is responsible for determining the female inflorescence in case of males, I mean to say. This is non-allosomic method. The third method is the hormonal method. Hormonal method of sex determination. Hormonal method of sex determination. In this case, I think the line tells us there is involvement of hormone, and this hormone will tell us about the sex. And this hormone is going to determine the sex of any individual. There are two very popular cases in this arena. There are very two popular cases. One of the cases, we call them as free Martin, and the second remarkable case is Crib's hand. I will explain them. One is the free Martin and second is the Crib's hand. What is happening in free Martin? Free Martin is occasionally we are witnessing. Occasionally it is being seen in case of cows. There may be a twin pregnancy. And this twin pregnancy will be dizygotic. Here we may have in this twin pregnancy, one may be male and the another will be the female. It may be seen in cows. Occasionally it may be seen in cows that the cow encounters a dizygotic twin pregnancy. One is the male, second is the female. And at the time of birth, what we see, the male is very strong, the male is fertile. However, the female is comparatively weak and the female is sterile. What has happened? What are the conditions which has turned the female sterile? It was in the womb, and when it was in the womb, it came out as a sterile female. What was the reason? Basically, it is the hormone. It is because of the presence of hormone that has made this female sterile. See what has happened. When we talk of these dizygotic twins, they are present in mother's womb, and there are vascular connections. Between male and female. By virtue of these vascular connections, what has happened? The male is developed very fastly. Sex cord is developed very fastly. And because of the development of sex cords, because of the development of testes in case of males, there will be the production of hormone known as testosterone, and this testosterone has been produced in the male, and because of this vascular connection, the testosterone from this side has migrated towards the female. It has migrated towards the female, and this testosterone has inhibited the formation of ovaries in females. It has not allowed the formation of ovaries in case of females. This testosterone has inhibited the formation of female genital tract or female primary sex organ in female. With the result, the females are born city royal. The second case I discussed is Creve's hen. See what is happening in Creve's hen. The Creve's hen is a famous case of sex reversal. It is the famous case of sex reversal. It was observed by Creve, and what he has witnessed, he witnessed a female, fertile female, a female hen, which was fertile. Fertile means the hen has given eggs, the hen has laid eggs, the hen has produced eggs, and after some time, there was sexual transformation in this female. Suddenly, this female turned out and reversed its sex and get converted into a cock. Oh my God, very strange. A female, fertile female, a female who has produced the eggs, has just reversed the sex. What is the reason? Basically, after investigating the matter, after investigating this hen, what he find this hen has basically developed a disease, and in that disease, she lost her ovaries. There was destruction of ovaries. And by way of losing ovaries, the hen has reversed her sex. She has reversed her sex. It means what ovaries were doing initially. Because once the ovaries have been taken out, the sex has been reversed. It means the ovaries are basically secreting some male inhibiting substance. Some male inhibiting characters are being secreted by normal ovaries, and with the result, there are no masculine characters in a female. The suppression of these masculine characters in the females are governed by the ovaries. And when ovaries got destroyed, the masculine characters were expressed, and that they were expressed in a form that the female hen has been converted into a cock. That's one more method. The fourth method, which we are going to discuss, is the haploid-diploid method of sex determination. Haploid-diploid method of sex determination. This method has been seen in case of Hymenoptera. Hymenoptera means it is seen in bees, it is seen in wasps, it is seen in case of ants. In case of bees, again, the method of sex determination is something very, very unique. What we see in bees, in the whole colony of bee, we have males, we have females, and all females are termed as workers. And we have one more thing which we call as a queen. We have a single queen. We have number of females, and these females are some are workers, some are protecting the web, and some are engaged with procuring the pollen. So many things, because there is a colonial life in case of honeybee. And there are few males large number of females and a single queen. Let us see what is the chromosome number. If we see, the males are always haploid, the females are always diploid, and the queen is also a diploid one. Males haploid, females diploid, queen diploid. Then, one more thing. The males are fertile. There's no problem with the male. The females, all the worker females are sterile, and the queen is fertile. Here, what we observe, the females are diploid, at the same time they are sterile. But it is the only queen among all diploid organisms, it is the among all diploid bees, only it is the queen which is fertile. Rest, all the females are sterile, and here the males are haploid but fertile. It means, who is determining the sex of the male? It is the haploid. Presence of haploid condition in case of male will determine the sex of male, will determine the sex. It's the male. And presence of diploid condition determines the female. Then there is one more separate thing, whether it is sterile or fertile. But the thing is that if it is a diploid, it is a female. It may be a queen or it may be a female. It may be a worker. But if it is a haploid, it is always a male. See what is happening in them. The chromosome number in case of the diploid chromosome number in case of honeybees is 32. Let us see how the queen mates. It has been seen queen mates at one time after nuptial flight. This queen, for example, this is a queen, this is a queen, it is diploid, the chromosomes are 32. And here we have a male, we have male sperms, we have a male, we can say the male has the total chromosome number of 16. The queen will show gametogenesis. Being diploid, it will show gametogenesis. And it will produce eggs. And all the eggs will be haploid. All the eggs will be haploid, having 16 chromosomes. Possessing 16 chromosomes. The males, since the male is haploid, there is no question of meiosis. There is no question of meiosis. Therefore, the males will produce the sperms. The males will produce the sperms by the mitotic divisions. By the mitotic divisions, not by the meiosis. By the mitotic divisions, not by the meiosis. And all the sperms will be again haploid. There will be no change in chromosome number because of mitosis. Female is laying the eggs, female is producing the eggs, and the eggs are haploid, but the males will produce the sperms, and the sperms are haploid. Since the males are himself haploid, they cannot undergo meiosis, they undergo mitosis and produce haploid, and they produce haploid sperms. These are the sperms. What happens? This female, once it mates with the male, it keeps a store of sperms in the receptacle. In the receptacle, the female possesses a receptacle. It keeps the store of all the sperms. Then, how many males and how many worker females are being produced will again be determined by the queen itself. If she wants to develop a diploid male, what she will do? She will fuse. There will be a fusion of the sperm and egg. With the result, it will be a female, a diploid female. It will be a diploid female. For example, if she has to produce a male, female will be produced by the union of egg and sperm. While as how a haploid male is born, the haploid male is born from the egg cell. From the egg cell, without making it to combine with the sperm, and there is a complete formation of male, haploid male, from a single haploid cell by the process of parthenogenesis. Parthenogenesis will take place and a male will be raised from a single haploid egg. this way. Now the question is, this is a female, diploids are females, and number of females are produced. Who is going to be the future queen? The question is, we have number of eggs we have, and all these eggs are basically diploid. But the question is, these diploids are females. Yes, we can understand, but females, we have two types of females. One is the worker and second is the queen. Who is going to be the future queen? There is only one queen. There is only one queen in the hive. If the queen left, or if the queen is old, then they can raise another queen. How? All the zygotes, all the young ones are fed with pollen. All the young ones are fed the pollen. But there is a particular young one. Which is being fed royal jelly. And that young one is the future queen. It means if we have to differentiate a worker and a future queen, or future workers and future queen, we can say it is the type of food. There is nothing genetic. In genetics of a worker and genetics of a queen is same. But what it determines? It determines a queen is one which will receive a special type of nutrition by way of royal jelly, and the workers will not get the royal jelly. This is the haploid diploid method of sex determination. Now the last and the most important method is the chromosomal method of sex determination. Chromosomal method of sex determination. See, the sex will be determined by the chromosomes. It is true in our cases also. It is the chromosomes which tell us the sex of individual. If somebody is XX, it's a female. If somebody is XY, it's a male. So chromosomes are telling us who is going to be the male and who is going to be the female. Here, I will tell you there are number of cases. There may be sometimes heterogamity in the male. There may be sometimes heterogamity in the female. I will try to simplify this by using this table. For example, I will write on this side I will write the females. This is the female, and on this side I will write the males. Okay, we have number of methods. The first method is XX and XY method of sex determination. The second method is again XX, and we have X0 method of sex determination. The third method I will write it again, XX and XX. I will write it with some other pen. It is XX. This is also XX. Okay. Then the third method we have, that is this way. It is, I will write here ZZ, I will write here ZW. And on the opposite side, we have ZW and ZO. They all are the males. They all are the females, and they all are the males, right? And at the same time, if we see in the first case, this is the first case, this is second, this is third, this is fourth. You remember this? You can write it this way: XXXX, ZZ. Sorry, it's not ZW, it is ZZ. I'm sorry. It is ZZ. Right. You can write it this way: XXXX, ZZZZ, XY, XO, ZW, ZO. Here, if we see who is going to be heterogametic, this is the female. Female has both the two chromosomes are same. Females are homogametic, but it is the male who is heterogametic. So we can say here we have male which are heterogametes. In this case, again we have the male which is heterogametic. In this case, we have the female which is heterogametic. And in the last case, again we have the female, which is heterogametes. It is male heterogrammatic in XXXY. It is male heterogrammatic in XXXO. It is the female which is heterogrammatic ZW. And here also it is the female Z0, which is heterogrammatic. And who is going to determine the sex? In our case, is this the female which is determining the sex? No, it is the male. It is the type of sperm which will be produced by male that will determine the establishment of male or female. It means those who are heterogametic, those who in all among these groups, those who are heterogametes will determine the sex of offspring. That means in this case, this is going to determine the males will be going to determine the sex of offspring in case of XX and XY. In case of XX and XO, it is the male again who is going to determine the sex because male is heterogametic. In ZW, ZZ, it is the female who is going to determine the sex in ZW, ZZ method. And in ZO, ZZ, it is the female again who is heterogametic and who is going to determine the sex. We can remember their examples also. In the first case, we have some most insects. Here the example is we have most insects like fruit fly, and we have human beings. We have the same method. We have human beings. In the second case, we have roundworms. And we have also in this case we have roundworms, and we have also some insects like butterflies, cockroaches, or we have grasshopper. In this case, we have reptiles and birds. And in the last case, that means ZO and ZW. In this, we have some we can say moths and some butterflies. These are the examples. If we see the crosses, what is the expectancy of receiving the male and female? In such cases, I will just discuss it. Let me take the first method, XX and XY, which is found in human beings. Let me take the XX and XY method. For example, if we talk of human beings, we have 44. If it is a female, it has 44 plus XX condition. And if it is a male, it has 44 plus XY condition. Males are heterogametic. Then gametes will be produced. The gametes will be having half the number of chromosomes. It is 22X. Again, half the number of chromosomes, 22X. Again, the gametes will be produced. It will be 22X and 22Y. See, what it indicates. The gamete produced is 22X. The gamete produced 22X. It means the females are homomorphic because they have the same X chromosomes. If the females possess the same X chromosome, same sex chromosomes, we call them as homomorphic. Here the females are homomorphic. And what we are witnessing, what we are seeing, the homomorphic females are producing the similar type of gametes, 22X. Those females who are homomorphic are going to produce homogametic, are going to produce the identical type of gametes like 22X and 22X. In case of human beings, the females are homomorphic and therefore they are subjected to produce similar kind of gametes and therefore they are homogametic. On the contrary, if we see the males, we have 44 plus XY. The males can produce the males can produce two types of gametes, 22X and 22Y. They are different. This gamete we call as gynogamete. Gynogamete. And this gamete we can call as androgamete. Androgamete. So males are producing two types of gametes, gynogametes and androgametes. This is the gynogamete, this is the androgamete. Let us see the crosses. For example, first of all, we'll make the cross between this and this. It will form 44X and X. Then this and this. It is 44X and Y. This and this. It is 44X and X. Then this and this. It is 44X and Y. Is this the female who is responsible for determining the sex? Female has given only one thing. It is 22X. Nothing else. We have this a female, this is a male, this is a female, and this is a male. It means it is the male. If the male produces gynogamete, the gynogamete will fuse with 22X and it will form 44XX, a female. So it is the male's gynogamete which is going to form the female, which is going to establish a female sex. And it is the male's androgamete who is going to unite with the female's 22X and is going to form a male. The blame is always being put on the females for giving birth to the female fetuses and are being considered responsible for giving birth to female gametes. However, the female is not. She is not at fault. She is always producing 22X. She is always producing 22X. It is the male who has a gynogamete and androgamete, and it is the male's heterogamete which is responsible for establishment of male and female. Therefore, we should never blame females responsible for giving birth to female or male fetuses. It is basically the father who is responsible for all this. And what is the sex ratio? Female, male, female, male. 2 males and 2 females. It is 1:1. The females and males are being born in equal ratios. The similar thing happens in other cases also. If we talk of any other example, it is XX and XY. Suppose we can say we have XX and XO. If we have XX and XO condition, XO means X0, X0. They are the females and they are the males, right? The females are again homogametic because they are homomorphic, and the males are heterogametic because they are heteromorphic, and therefore they can produce XX, they can produce X0, XX, and X0. This ratio is 1:1. Everywhere it exists. Here, whether this is the first method, whether it is XX and XY, whether it is XX and XO, whether it is ZO and ZZ, or whether it is ZW and ZW and ZZ. Everywhere, whosoever is the heterogametic, whosoever is the heterogametic will be responsible for establishment of sex, will be responsible for determining the sex, whether it will be the male or whether it will be the female. That's all about this topic, and in the next video lecture, I will tell you about the chromosomes. What are the basic things which we need to know about the male and female sexes, X and X and X and X, X and Y. On that basis, we'll be able to understand all those diseases who are X-linked, all those diseases who are XY-linked, and all those diseases who are Y-linked. Thank you very much for watching.